Hi everyone, my name is Pablo Martínez Cañada and I'm going to present the topic Biophysically Detailed Cortical Circuit Modeling in Aging Research. Understanding how brain signals measure particular patterns of neuronal activity depends on forward models and inverse models. Forward models describe how the recorded brain signals are generated by neuronal activity. Inverse models are used to analyze the brain recordings and infer the underlying neuronal sources. Inverse modeling is a complicated task because it is an ill-posed problem, which means that different patterns of neuronal activity can generate the same electric or magnetic signals. A powerful approach to deal with the inverse modeling problem is to extract features or biomarkers from macroscopic brain signals and link these features to parameters of forward models. As we will see later, we have identified specific features, for example the 1/f spectral slope, that potentially index the underlying change in the synaptic excitation inhibition ratio. This biomarker was used, for example, to demonstrate that EI imbalance affects autistic males and females differently. Also to uncover different regional patterns of EI during Alzheimer's disease progression. Previous models in aging research have simplified description of cortical dynamics to just a few variables that represent population average activity. Neural mass models and neural field models have been proven useful to explain large-scale neuronal processes and structure-function coupling at macroscopic scales. However, their ability to make precise biophysical predictions on cellular and local circuit-level processes is limited, as the parameters are an abstraction representing population-level activity. On the contrary, our research relies on spiking and multi-compartment models to study the relationship between neuronal and synaptic changes mediated by the local circuit with global dynamics and properties of field potentials. There is substantial evidence regarding changes of local properties in the cortical circuit that potentially underlie a spectrum of clinical conditions, so we believe that a biophysical detailed circuit model is necessary for such an application. Cortical circuit models may account for different levels of detail, ranging from biophysically detailed multi-compartment neural networks, top level, simplified spiking point neural networks, middle level, and population type models accounting for population average activity, bottom level. The different levels may at times be bridged with appropriate mapping of parameters. Multi-compartment models may be directly combined with volume conductance theory for extracellular signal predictions, as these models account for the spatiotemporal distribution of transmembrane currents. However, we cannot compute the extracellular signal predictions directly from spiking point neural networks or population models. They require some additional method, here illustrated by the black box, for translating spike events or spike rates into representative extracellular signal approximations. Over the years, we have developed robust methods for computing accurate brain signal predictions, and we have validated these methods on both simulation and empirical data. In this first study, we combined in silicon modeling, in vivo chemogenetic manipulations in mice, and brain scans of people with and without autism to demonstrate that specific biomarker, the Hurst exponent H, can be a robust biomarker for excitation inhibition imbalance. We first simulated a spiking circuit model and found this relationship between H and the synaptic EI ratio in this model. As you can see, when H is reduced, there is an increase of excitation. We then explore which types of manipulations of parameters of the spiking circuit model can approximate the effects of in vivo chemogenetic manipulations in mice. We capture effects of the different classes of chemogenetic manipulations by changing the voltage threshold for spike initiation and the resting potential. Finally, in autism, we found that H is reduced, indicating increased excitation in the medial prefrontal cortex, area P32 of autistic males, but not females. In this other study, using the aperiodic component from resting state spectral data and computational modeling, we developed a new biomarker for inferring EI alterations in Alzheimer. We first found that aperiodic 1 over f slopes computed from model simulations decrease, which means flatter slopes, with increasing EI ratio. We next analyzed EEG data from an open dataset that includes resting state EEG recordings of healthy individuals across different ages and patients with Alzheimer. First, we observed a flattening of power spectrum slopes in older individuals compared to the younger group. To test whether slopes can be used to predict changes in EI, we computed 1 over f slopes of individuals in Alzheimer and control groups and used the brain model to compute predictions of EI imbalances. The topographic representation of changes in EI at the bottom revealed an interesting pattern in Alzheimer with opposite effects, suggesting a coexistence of excitation and inhibition dominated regions. Indeed, when comparing local differences in EI across subsets of electrodes, we found that, for example, temporal electrode locations, such as T6, displayed a significant shift toward inhibition. In a current project, we are using a database, the POCTEP database, which includes EEG recordings of healthy individuals, 51 controls, and patients with Alzheimer's disease across different stages of the disease, mild, moderate, and severe. This database was shared by the Biomedical Engineering Group of University of Valladolid in Spain. We computed features from EEG data using the Catch22 library, which includes 22 feature-based representations of time series. These 22 features were selected by the corresponding authors based on their classification performance across over 100 time series classification datasets. In our work, we analyzed these features using a statistical test, such as linear mixed models. In this plot, we show the effect size for this test, and we found a very interesting result here. Some features, such as DFA, show opposite effects during progression of the disease, while other features have an uniform change. We have also simulated a computational model and computed the same 22 features. In this way, we could link changes in the EI ratio with changes in these features, as we did in previous works. Remarkably, we found that predicted changes in EI inferred from features such as DFA can be associated with regional patterns of imbalances in EI ratio in Alzheimer. So the expected contribution of our work is a set of methods that can uncover clusters of biomarkers and can guide future research of biomarkers in AD. We will first perform massive feature extraction from brain recordings using a large collection of time series feature libraries, such as Catch22. We will then identify the different clusters of spatial patterns indexed by the different features during progression of the disease. So in this way, feature studies in Alzheimer can use these results to decide which biomarkers are the most convenient for exploration of changes in the cortical circuit mechanisms. So to sum up, this is the whole picture of our research project. First, we simulate artificial brain signals from biophysically detailed brain models. We then use databases of time series brain recordings, such as EEG or MEG data. In this first stage of the project, in which we are now, we have performed massive feature extraction from real and simulated data and compared these features. And in the next stage, we want to use machine learning algorithms such as simulation-based inference, SBI, to automatically predict changes in cortical circuit parameters from non-invasive brain recordings. So this brings me to the end of my presentation. Thank you so much for your interest and attention. And if anyone has any questions, I would be more than happy to answer them. Thank you very much.